Okay, so today is Muscular System Day 2. Today we're going to be learning about the sliding filament model, muscle proteins, the different types of muscle fibers. So there's like fast and slow twitch fibers, which we'll be going deeper into. We'll also be talking about muscle contraction, action potentials, and the summation model. All right, so let's get started by talking about what a sarcomere is. So last time we talked about how the structure of a muscle fiber how each muscle fiber makes up a fascicle, and each fascicle makes up a whole muscle. So today we're going to be going even smaller, and we're going to be looking at each individual muscle fiber. And if you recall, a muscle fiber is just a muscle cell. So we're just going to be looking at one repeating unit inside each. We're going to be looking at each repeating unit inside each muscle fiber. So this right here, one of these is a sarcomere. And so like I said, each sarcomere is the repeating unit in the striated muscle, and that's what gives it the dark and white stripes. And so each sarcomere connects. Can I go back to the first slide? I'll be referring back to it. Yeah. So but a myofibril is the contractile organelle of skeletal muscle. So we have myofilaments, and there are also myofibrils. And what the myofibril is made up of many sarcomeres connected together, like hundreds and hundreds of them. All right, so the cross-bridge cycle is the mechanism where the myofilaments slide over each other during muscle contraction. And that might not make any sense right now, but we'll be talking about what myofilaments are. And so today we'll learn about how muscles contract on the molecular scale. Yeah, so for your note sheet, it's the sarcomere is the repeating unit in muscle tissue that makes up a myofibril. And the myofibril is just the contractile organelle of the skeletal muscle. Okay.
Repeat that again.
Wait, but what about the first slide? Does that have that doesn't seem to have anything like
there's nothing here that's needed on the note sheet, but it's a very good diagram, so I'll be referring back to it because it'll help you guys like visualize what's going on.
Okay, thank you.
Yeah, no problem. Okay, so if we look here to the image on the left, this is the simplified diagram of what a sarcomere is. And yeah, it looks weird because this is not what it actually is like, but it helps you to visualize each sarcomere. So I'm going to go back to this first slide real quick. And as you see here, in each muscle fiber, there's like repeating sarcomeres. And so this diagram represents one of those. So I'm just going to be talking about each component because they're pretty important when we talk about muscle contraction. All right, so first we have our Z line here. So the Z lines are these discs, and they're like plate shaped, and they're made of dense protein material, and they separate each sarcomere from one another. So from one Z line to the other Z line, that'll make up one sarcomere. Next we have the A band here, which is in the middle, and it's just this darker middle part, and it represents the entire length of these thick filaments here. So I guess I have to talk about what thick and thin filaments are first. So so a thick filament is basically, it's made of myosin. So I'm just gonna go here real quick to talk about what a thick filament is, because the sarcomere diagram won't make sense until I talk about what those are. But each sarcomere has these repeating lines, as you can see. And so these are the thick and thin filaments. So is anybody confused right now? Because I can go back and.
Yeah. I don't get it.
All right. So let's talk about the myofibrils again. So within each myofibril, which, as you recall, are the contractile part of the muscle cell, they're made of two different types of filaments. And so these filaments are just basically proteins in lines. So as you see here, these thin lines are called thin filaments, and these thicker lines here are called thick filaments. And so in a sarcomere, this is the thick filament and that's the thin filament. And what happens is they're going to slide over each other during contraction, and that's what helps your muscles to contract. Yes, thin things are thin and thick things are thick. So once we are good on that, we should learn about the different.
Thin filament again?
The thin filament, you can see here, they're located on the outer edges of the sarcomere, while the thick filament's in the middle. And don't worry if you don't get
the diagram on that, like that slide, it doesn't look, I think, no, not.
Yeah, I know. The one on the note sheet, right?
It barely looks like, yeah.
Yeah, so I put that different diagram there, hoping that once I teach you guys what these parts are, you could look at a different diagram and know how to label that one as well.
Oh.
Yeah, but don't worry. We have like a ways to go before you guys have to fill that in. All right, so hopefully you guys are good on what a thin and a thick filament are, and we'll be like solidifying that later. But just know for now that we refer to the different sections of a sarcomere with different names just to help us visualize the contraction process better. So just know that the I band is the section of the sarcomere that contains only thin filaments. So you know how striated muscle has light and dark sections here? So whenever there's a thick filament, that's what gives it dark.
Slide for a second.
What?
Can you go to the previous slide? Yeah, thank you.
I have a question.
Yep.
What's the H zone from the next slide?
Okay, so what the H zone is the part of the sarcomere that contains only the thick filaments but not the thin filaments. So if we look at this picture of a sarcomere, if you see where my mouse is, do you see this part where there are no thin filaments but there's only thick filament?
But I thought that was like the A band, the part where the thick filaments are. And
yeah,
then
so the H zone, the H zone is in the A band. So it's not like the A band this is like only the A band here. So the center of the A band we call the H zone because it's still where thick filaments are, but now there's no thin filaments. Does that make sense?
yeah, because it's like the H zone is the empty part where there are no thin filaments.
Wait,
but there are thick filaments.
for the note sheet, how do we put in the numbers? There's the numbers.
I'll go through it with you guys. So
okay.
Yeah. All right.
You don't need the numbers.
Oh.
And also, I have one more question. What do you mean, when our muscles contract? Do you mean you mean move?
Yeah, when they move. So what happens is to make you know how when you contract your muscles get shorter. And that's basically the reason why that happens is because these thin filaments and these thick filaments in a sarcomere, they're going to overlap with each other. And I'll show you guys a video that'll make that a little bit easier to understand. But during contraction, they slide over each other. All right, so I'm just going to go over what the I band, A band, H zone, and Z line are. And seriously, don't worry if you don't understand this. the first time you're learning it because it's pretty complicated. So like I said earlier, yes, the red parts are thick filament and the green is thin filament. And so, like I said, the Z discs are what separate each sarcomere from another, and they cut in the middle of each thin filament. The A band is this darker middle part here, and it represents the entire length of the thick filament. Next, we have the I band, and the I band is basically anything that's not A band. And so notice how in the I band we only have thin filaments, but there's also the region here within the A band where there still is thin filament. So like the thick filaments and the thin filaments are naturally overlapping just a little bit, but the part of the thin filament that extends into where it's under the thick filament, we still put as part of the A band, which is the darker part. All right, so that might have been a lot, but we'll come back to this. And then, like I said, the H zone is this part in the middle of the A band where there are no thin filaments, it's just thick filament. Okay, I'm gonna move on. I know you guys probably still have questions on this, but we'll come back to it. So this is just a table for you guys to read over on your own time when I share the presentation with you. If you want to take a minute just to read over it right now to make sure that you're good on that.
I have one last question.
Yep.
So, you know how I understand like everything, but there was no M line in the diagram. Can you explain more what that is?
Oh yeah, sure. So there's no M line in this diagram. I think it's just because it's like this diagram is a little simpler. But what an M line is there are always going to be a line down the middle of the A band or the H zone. And so it's like in the center between two Z lines, and in here it's just like a line of supporting proteins and they hold everything together. So I think I have a diagram. Sorry, I'm going to go through here, but so do you see here how there's like a line in the middle down here? That's our M line. And so it just basically helps us to hold everything together. And it's called the M line. It kind of stands for like the middle line. Yeah, it's a lot. Okay, so I'm gonna move on. Do you guys remember what myofibrils are? Can someone tell me what a myofibril is?
Contractile organelle of a skeletal muscle.
Yeah, okay, so myofibrils are inside muscle cells. And so when we talk about a myofibril, they're built from 3 different kinds of proteins. So we have contractile proteins, and the contractile proteins are the ones that help actually make the contraction happen. So let's talk about those first. Has anyone ever heard of myosin or actin? So myosin and actin are both motor proteins. And what that means is they help different parts of our body, like inside our body, to move. So let's take a close look at what a myosin molecule is. So myosin proteins make up thick filaments. So when you think about thick filaments, I want you guys to know automatically that that's myosin. So myosin only makes up thick filaments. And so each myosin molecule, it kind of, you see here, kind of looks like a golf club. And so each myosin molecule is shaped like two golf clubs kind of twisted together. And if you see like the bulbs here, these are the projections, we call those myosin heads. Okay, so let me repeat that. Each myosin molecule is one of these, if you look at my mouse. And so these little projections here, they're called myosin heads. And what they do is they bind to the thin filament, which I'll be going on into the next slide. Is everyone good to move on?
Yeah.
Okay.
Hey, no, can you actually go over that one more time? Because I didn't understand it.
Nope, we didn't finish the 3 types of proteins that make up a myofibril. You want me to go back?
Like, over this again.
Oh, sure.
Thank you.
Okay. So myofibrils, as we recall, are made up of sarcomeres. And so each sarcomere has different types of proteins inside it. So myofibrils and sarcomeres are built from 3 different types of proteins. And one type are called the contractile proteins. And what they do is they help the sarcomere and the muscle overall to generate force during contraction. And so the first example of the motor protein is called a myosin protein. And so we have lots and lots of myosin proteins here, and they kind of lie on top of each other, and they like form a chain, and that's what makes up the thick filament we were talking about. And so what myosin does is it wants to bind to actin, and actin we'll be talking about on the next slide, but it binds to the thin filaments. So just know that the myosin heads are what help the thick filament to bind to the thin filament.
Okay.
Yeah. Okay. So our second contractile protein is called actin.
Back to the other slide.
Okay. Okay. Yeah, these are in skeletal muscle. Yep, myosin is an example of contractile protein. Okay, I'm going to move on, but as always, the slideshow will be posted right after class. So if you need to go back to it, I will be posting it. All right, next we have actin. And if you guys see like this line of yellow spherical things in a line, each one of those spherical things is an actin protein. And so we have tons and tons of those joined together, and we get this kind of helix looking thing. So ignore the orange things and the green things. We're just looking at the yellow chain of spherical proteins. And so this right here is what makes up a thin filament. And so each actin molecule is pretty small, and it joins to other actin molecules, and they twist into this helix shape here. And next, each actin molecule has a region where the myosin head from earlier really wants to bind to it. So that's what helps the myosin or the thick filament to attach to the thin filament. And that's going to be important later for when we want to have the contraction, which is where the thin filament and thick filament slide over each other to make the muscle shorter.
How does that actin get like twisty shape again? Like all of the actin beads stuff.
Sorry, can you say that again?
How does all of the actin beads make like that twisty shape? How does it get into that twisty shape again?
That's how they, so each like actin, so you're asking why is it in a helix shape and like why they join together?
Yeah, like kind of like that.
I'm not sure why it's in a helix shape. If Jos or Sanvi know. But the reason why that it's like in a chain is because like each like actin molecule is like powerless on its own, so we need to have them in a chain in order for it to make up a thin filament. And I'm not sure if that answered your question, but if you want to like, I think I can probably help look up the answer after class.
I think.
Yeah. Okay, so the next category of proteins that make up a myofibril are the regulatory proteins. So that might be on your note sheet, so you might want to write that down. And so what the regulatory proteins do, they help to control whether the myosin molecules are binding to the actin molecules. So the first molecule here is troponin. Troponin is like that green protein we see, and it's responsible for binding. It's responsible for preventing actin and myosin. So what troponin does is it has many different functions. But so one part of troponin is going to bind to this other protein called tropomyosin, which we'll be talking about in the next slide. Another part of troponin binds to actin. So troponin actually, if you guys remember, we were talking about that site or region on actin that binds to the myosin head at resting state. Troponin actually prevents actin from binding to myosin. Finally, we have the last part of the troponin molecule, and it binds to calcium ions. And so I'll talk about calcium ions a little bit more later, but just know that calcium ions are really important for starting muscle contraction. And so if you're wondering what these T, I, and C letters mean, the troponin protein is made of different parts, and we call them subunits. And each subunit has a letter as its name. So we have a T subunit, an I subunit, and a C subunit. And all of them together make up that troponin protein here. Is everyone good to move to the next slide? Okay, I'm going to move on. Last type of regulatory protein is called tropomyosin. And like we said earlier, tropomyosin is bound by troponin. And so what tropomyosin does is it also helps to cover up those myosin binding sites on actin, so myosin can't bind to actin. Troponin, on the other hand, when our muscle gets a signal that it needs to contract, troponin will move tropomyosin away from those actin binding sites. And then our myosin heads can bind to the actin. Okay, moving on. Our last type of protein that makes up a myofibril are called the structural proteins. And so they don't contribute directly to muscle contraction, But what they do is they keep our sarcomere in the structure we want. They link each Z disc and M line together. They just allow us to keep our filaments in the right alignment. And so the most important one of these structural proteins is called titin. Titin, as you can see here. So can someone tell me what this blue thing is? Just like briefly moving aside from what this is right now, but can someone tell me what the blue thing is? Or does anyone think they know?
Thin filament.
Thin filament.
Yeah, that's a thin filament. And so you know because, well, it's like thinner, but also because you don't see it in the middle of the sarcomere. And so does anybody know what this is? If you see my mouse.
The thick filament.
Thick filament.
Yeah, that's a thick filament. And you can see like the little bulbs coming off of it. Those are the myosin heads. So this is like slightly zoomed out. But if you recall, yeah.
I thought that the myosin heads were purple, though.
What?
Didn't in the last slide didn't it say the myosin heads were purple?
Oh, well, those are just diagrams. In our body,
Okay.
they're not colored purple. Yeah.
What color are they actually in our body?
I actually don't know because when we look at proteins, like when we look at the pictures of proteins, they're usually like stained. So I want to say they're like probably like, yeah, I actually have no idea what color they are. And it's like it's not that.
Everything in the diagram is red.
Hmm?
I think everything in the diagram is red. Like it would be red in the actual body.
That's okay. I guess it doesn't really matter like what color they are because like proteins are so microscopic that I think when you look at them, they're probably not even gonna like have any color because like the parts of the like muscle cell that give it its color are not found in the sarcomere here. I don't know, you wouldn't even be able to see it. Okay, anyway, this is the Z line or the Z disc. We can call them either disc or line, it's the same. Here coming down the middle is that M line. It's like M for middle. And they've also labeled the I band and H zone, and this would be the A band. Yep, okay. So back to titin. Titin is, if you can see here, that line that extends from the Z line to the end of the thick filament is a titin molecule, but titin actually connects from the Z line to the M line. So even though you don't see it in this diagram, titin is really, really elastic, and it can stretch to 4 times its size and then spring back to the original size. And so if you recall from last class, muscle is really stretchable, and we call it like elastic and extensible because it can return back to its original size. And that's thanks to our friend titin, because when the sarcomere will contract, titin is responsible for causing the muscle to spring back.
Also, fun fact, titin is the largest possible protein. So
Yeah.
the thing is, proteins are made up of like these different chains, but sometimes they're like bonded together to form different like proteins connected together. But titin is one single protein, but it's very, very large.
Yeah. Okay. So before we do this slide, I want to show you guys a video. Hopefully this will work over Zoom because I know sometimes like videos are kind of laggy. So if it is laggy, try to like see if you can, like if it's like so laggy that you can't even understand what's happening, let me know. but otherwise, just like try to watch it. So,
You use muscles every day to do activities.
how is that?
This woman is using muscle.
Can you
Good.
good? Okay.
It's not like it all so far.
Yeah,
It's not even laggy.
Circulate blood and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is
You guys remember the neuromuscular junction from Tuesday, right?
Yes.
Yeah. So like
Yeah.
how acetylcholine, yeah, and everything.
where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance.
Yep. Oh wait. Oh, sorry. I don't know how that happened. Whoops. Oh no.
You use muscles and exert force on them to move,
Sorry.
called the M line.
So do you guys see? Oh my God, why is it doing that? Sorry. Oh, okay, I think I was trying.
You use to one
Okay,
another.
so do you guys see here how that's like there's light and dark stripes?
Yeah.
Yeah, so that's those are like the striations, and you can see it here a lot more clearly. But where the thick filaments are is where we get that darkness, and where the thin filaments are, we have that lightness.
Down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force.
Okay, so I didn't teach you guys about what cross bridges are because I didn't think we would have enough time to. So if you don't get what that part is, it's fine. Just notice here how the myosin heads are binding to the actin and pulling it
on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium.
Okay, so this part is important. So pay attention to why calcium is important.
The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows...
Okay, did everybody get that? So
But
calcium,
not really.
Yes.
I'm gonna replay that because it's important.
Yeah, there was noise in the outside.
Okay.
ATP is present, calcium
So don't worry about ATP. Just see how calcium is binding to troponin.
ions bind to the troponin, which displaces tropomyosin, exposing the
So did you see how like the pink molecule lifted kind of once calcium bound to the troponin? Yeah, so that's how we get the tropomyosin to expose the actin binding sites.
myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores.
I'll talk about that
Calcium
next.
ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, so I hope that made it a little bit clearer how the sliding filament model works. Yeah, so like,
Wait, you post the recording every day in the classroom, right?
yeah, I will.
Or did you? Oh,
I
okay.
hope that it made it a little bit clearer, just how you guys can see how each individual sarcomere sliding over and overlapping causes the whole muscle to contract. So that's the big picture thing I hope you got from that. Okay, so the video talked a little bit about T tubules and the sarcoplasmic reticulum, so I'm just gonna talk about that real quick. Okay, so this is a picture of a muscle cell. It's a good cross section. And so the first thing that you might see are the transverse tubules or the T tubules. And so if you see here, this green thing is the plasma membrane of the cell. And so what the T tubules are they're kind of like, they kind of like tunnel in from the surface of the cell, from the membrane, and they go into the center of each muscle fiber. And so why they're important, because when your nerves send that signal to your muscle to contract, the T tubules help to make sure that that signal gets everywhere in the cell. So since they're like T tubules that regularly occur, they just make sure that that signal is exciting all parts of the muscle fiber at the same time. So just know T tubules carry this signal to contract. Next is the sarcoplasmic reticulum. And so what that does is it stores the calcium ions. And remember that the calcium ions are really important because they allow for contraction when they bind to the troponin molecule. So when the sarcoplasmic reticulum gets a signal from the T tubules, it releases those calcium ions. And when the calcium ions get released, that's when your muscles contract. And so these pink things are the sarcoplasmic reticulum. Finally is the sarcoplasm. And if you guys know what cytoplasm is, sarcoplasm is just the muscular cell version of cytoplasm. And it contains two important things, glycogen and myoglobin. And so that will be talked about in the next slide. So is everybody good on that? Does anybody have any questions? All of these things together, the myofibril, the T-tubule, and the sarcoplasmic reticulum, they all work together to help contraction happen in a muscle cell. And see here, there's like many myofibrils inside each cell. Like here, there's like 6 of them. And they all are made of repeating sarcomeres. Okay. Why are there 79 messages in the chat? Okay.
Are we gonna hoot today?
Yeah, there will be a Kahoot today.
Every single time we do a Kahoot, can you like post the links for the Kahoot so we can practice it later?
Yeah, I will. Thanks for reminding me. Okay. So the next thing we're going to be talking about is the two different types. So this is now we're moving away from like the whole sliding filament model and contraction and everything. Let's talk about two different types of muscle fibers. So not all skeletal muscle fibers are the same in the way that they function and what they're made of. So we categorize them broadly into two different types: fast twitch muscle fibers and slow twitch muscle fibers. So skeletal muscle fibers that have lots of myoglobin, and myoglobin is this red protein, and it's responsible for collecting all that oxygen that's in our blood and making sure that it's going to be used in our muscles for energy. So slow twitch muscles are, as you can see here, they don't fatigue easily, and so that we find them often in like endurance, people that do endurance sports like marathon running, and they're found in like the different parts of your body that need to be able to like not fatigue easily as much. So can anybody like shout out where they think some slow twitch muscles might be found in your body?
Like
Neck.
Yeah, definitely neck, because it needs to be like holding up your head all the time, right? So it really doesn't need, it really can't fatigue easily.
Legs.
Your what?
Legs.
Legs? Yeah, that's definitely true.
hip,
Your arms,
hip,
Your ankle.
your hip.
your arms,
Your
because like you need to like move your arms up and down.
So this is actually a trick question because there's slow twitch muscles like everywhere in your body. It's just that some limbs in your body have more slow twitch muscles than fast twitch, like more slow twitch than fast twitch. But you'll basically find slow twitch muscles everywhere in your body. But you're right, like for like your parts of your legs that like are responsible for like running more. Okay, so like when you're sprinting, okay, sorry, let me rephrase that because that might have been confusing. But it's kind of like when you're sprinting, your fast twitch muscles are going to be used a lot more, and when you're like running a marathon, your slow twitch muscles are going to be like more we're depending on them more to work. So these are the slow twitch muscles, and they're dark red, like I said, because they have a lot of myoglobin. And so that helps us to have a steady source of energy. Slow twitch muscles are also smaller in diameter, and they don't fatigue as easily. They have a large blood supply, and they use oxidative phosphorylation as their primary energy source. And I don't think we're going to have enough time to like go into what that is. So if you're interested, you can research that on your own time. But basically, it's just that it's a way of generating energy that uses oxygen. So you should just know that it uses oxygen to make energy. Next are our fast twitch fibers or fast twitch muscles. They use a type of, they use a process that doesn't take oxygen to make energy. They're larger in diameter, they use less oxygen, and they fatigue a lot more easily. So yeah, you'll need to use your fast twitch muscles when you're sprinting. Can anyone think of like any other activities that might require your fast twitch muscles to be active? Yeah, I think these are all good, really good answers.
When you touch a light bulb, you react to it by
using the brain that quickly.
Mm-hmm, yeah. Okay. This table has all the important like differences between slow twitch muscle fibers and fast twitch muscle fibers. So you guys can go back to that on your own time to copy it into your notes if you need it. Just notice here how these slow fibers are like so much darker than those fast twitch fibers. And you know how like we call meat red meat and white meat? That's actually due to like the composition of there being more slow twitch fibers or fast twitch fibers. So that's pretty interesting. Like chicken breast is like white colored, and that's because it's made of more fast twitch fibers. Okay, last thing today before we go to the Kahoot. So, a twitch is a single muscle contraction. So a twitch contraction is basically the contraction of all muscle fibers in a motor unit when one signal arrives at the muscle cell. So all the muscle fibers in a single motor unit contract, and that's a twitch contraction, just one contraction. But that's like only like if you like flex your bicep really quickly and then let go. But the next thing is called wave summation. And so that's like when you're like contracting your bicep for a lot longer. So maybe like when you're lifting weights, it has to stay contracting for a much longer period of time. And so what happens is if a second stimulus from the brain comes before your first contraction's over, the second contraction is going to be way stronger. And so this is a kind of complicated diagram, but as you see here, one row is showing what's going on when you're contracting your muscle, so like the tension. And the second here on the bottom is what's happening behind the scenes, like how the signals are coming to your muscle. And so this, when your muscle is contracting at the highest tension, there's going to be tons and tons of signals coming from your brain, just like one after the other, after the other, after the other. And so, oops. So that means that the muscle will never get a chance to relax. And so it just continues to stay contracting. Okay, so I'm sorry if I had to like rush through that last slide, but I just want to make sure that you guys had enough time to do the Kahoot. So that was today. I know today was like a lot of new information, so definitely go back and review the material that you might not be solid on. Okay, so everybody pull up kahoot.it.
What's the pin?
I think it's coming.
Okay.